Okay everyone, today I'm gonna present you the Trapdoor spider life history through slow science. So, what is this topic all about? This topic is all about understanding life history like demography and biology of Trapdoor spider by using slow science method to overcome the conservation issue. Before we start, I'm going to tell you about the biodiversity loss that be the major problem in our ecosystem. What is biodiversity loss actually? So biodiversity loss is declining of some species number in ecosystem. Like you can see over here is some already extinct species like dodo, passenger pigeon, and Tasmanian wolf. They already extinct back in the time. And what is the effect of biodiversity loss? The first one is the ecosystem disturbance. As you can see in here, there nothing left. Everything is screwed in the environment. And the second one is the biodiversity loss because of hunting. You can see over here is the Javan tiger that already extinct because of over hunting. And the third one is decreasing in natural resources. Biodiversity loss develop the conservation in biology, and the conservation in biology is mainly focused on the maintaining the species diversity. And I'm gonna tell you, the fundamental aspect is to contribute and maintain the process, so the endangered species could survive. But sadly, most of the conservation effort is only focused on some vertebrate, like you can see over here, like the conservation for the African elephant, tigers, and many vertebrate, the endangered ones. But actually, invertebrate had the same effect due to biodiversity loss. Especially arthropods, they are more diverse and had a lot of eco-function. This small arthropods has little to known life history, and that would make it more difficult because in order to do some conservation effort we have to know how they live. For example, in over here is Bombus avinis. They pollinate 65 different genera of plants, and they are endemic to North America. That's really important. And over here is Capnia lacustra, an endangered aquatic insect that endemic to Lake Tahoe, and they are associated with deep water plant beds, and it's also important. And to address that kind of problem, slow science method had been developed for many years to solve it. And before I'm going deeper into this topic, I'm gonna introduce you to the person behind the slow science. She is a Barbara Yorkman. She has done 41 years of continuous research in slow science. 41 years. That's that. Uh, that's a really long time. She is a conservationist and arachnologist who promotes slow science method to study these particular spiders, the trapdoor spiders, Gaius velocis. And here we go. There are three questions according to the slow science. There's what is slow science and why it has to be slow science. What is the benefit? The first one is what is slow science? Slow science is a research method that help us to understanding the life history of the species from time to time, time to time. The key word is that from the time to time and progressing along period of research. And why it has to be slow science? Because scientists should not expect to provide quick fix to the problem. Um, Science is a marathon, not a sprint. It's from Owens 2013. And what is the benefit of slow science? First, it gives more variable research item, and second one is it gives more precise information to the public, and the third is independency. This 3 point is important and it's not related to time it's not limited by time. I mean, and here comes the main objective of this research. It's to understanding the biology, growth rates, lifespans, and natural history of Euplus grandis by slow science to overcome the conservation crisis. And this is the spider that we researched, and the name is Euplus grandis, as I mentioned before. This is a new species of trapdoor spider, and this species is only found in eastern part of Australia and recognized as SRE species or short-range endemic species. And this spider is the biggest of all Euplus species, and this spider needs further research about his species life history to overcome their conservation issue. For the diagnostic character of this spider, as you can see in here, this is the morphological appearance of the Euplus grandis, and this is the male's palpal emboli that used to transfer the sperm into the female spermatheca. Usually the female is having a lot more distinguished feature from its own species, and both the female and male of Euplus grandis were bigger than any other species in their genera. For the methodology, we use site survey and study site survey, burrow locating, tagging, census, and data analysis. For the study site survey, its study site range is within the known of distributional range of this particular species in Brigalow Belt area. It's located on the eastern part of Australia, as you can see in the map. And for the burrow locating, the first burrow were conducted in September 2016 with systematic approach. The burrow were detected by the contrasting color of door decoration. Over here you can see it's very contrasting with their surrounding environment. It's covered by moss and covered by lichen, except for the D. This is like blending with their environment, and it's rather hard to recognize if it's a spider burrow. And for the tagging method, the tags were made from anodized metal, so it's more durable against the weather effect. And the tags were hammered into the ground 5 centimeters from the trapdoor. You can see in here, this is the trapdoor and this is the tag, and the tip of the tag was aligned to the burrow direction. This is the tip of the tag and this is the burrow. It helps to know where's the exact location of the burrow, so we didn't lose the burrow completely. This is the shape of the tag. It's an elbow dimension. As you can see in here. In total, there are 69 burrow tags in September 2016. For the census methodology, during the census documentation of each burrow, we check and open the burrow as assumption that active burrow contains single living spider and defunct burrow are evidence of mortality. We got an example here is defunct burrow, and it's typically open or partially collapsed, and they didn't have a fresh silk lining, indicates there are no active spider in here. And for the data analysis, to track the baseline growth rate and longevity, door diameter of each individual burrow were plotted over the time. This data form the basis of demographical assessment will useful to estimate the average growth rate and longevity. For the classifying the burrow, actually there's a 3 category. The first one is the smallest one is the younger juvenile, or we call it sling for the baby spiders, and the slightly bigger one is older juvenile including adult males, and for the biggest one is the adult female. There is no specific category for the spiderling or baby spider, since they usually live together with their mother as the proof of maternal care. For the results part, actually four um, there's a four different study. The first one is study set characteristic, census data, growth rate, and survivorship. For the study set characteristic, this is the example of their living environment of the spider. It's dominated by the Bragalo tree, Acacia herbaphila, and it's mostly dry with dead leaf scattered around the forest, and it's rather a right condition. It's very dry. For the census data, the 1st September 2016, there were 69 tagged burrow, including 41 adult females, and 18 older juveniles, and 10 young juveniles. The 3 older juveniles shift into adult females category, and 1 juvenile shift into old juvenile category. And per March 2018, 45 active burrow were being monitored, and it consists of 31 adult females, 12 older juvenile, and only 2 young juvenile. The census was complete every 6 months. With total, there are 3 census visits in March 2017, August 2017, and March 2018. For the each census, the presence of spiderling within maternal burrow is also noted, like the far right in here, as you can see some baby spider in one burrow. Besides, the door diameter will also note for measuring the growth rate. Taking the diameter of the block door at its widest point, like the middle picture, this is from the farthest left point to the farthest right point to count the diameter. The burrow diameter is correlated with the spider size, according to Vincent research in 1993 in other trapdoor species. And we also recorded that the spider is able to repair the trapdoor, and this is the example of the process. It's indicated that it's an active burrow because once the door gets destroyed, the spider immediately will repair it. As you can see, this is the broken trapdoor, this is their actual burrow, and the last one is the final installed new trapdoor. And we also note the growth progress of the lichen on the trapdoor surface over the month. You can see in here there are some black spots on the trapdoor burrow causing of the lichen, and over the time the black spots were actually increased, and it's the proof that the lichens were able to grow in the trapdoor. And for the survivorship, the mortality was highest among young juvenile spider. It's counted in 29% survivorship over the 6 months. And the mortality decreased once burrow reached the older juvenile age with 85% of survivorship. It's very high actually. And similarly, mortality of adult female was also low at 88% of survivorship, and it's increasing from 85% to 88%. Their life expectancy also increasing. For the growth rate, although the growth rate and average growth curve are difficult to calculate after only 1.5 years, but some sample indicates that the immature spiders spend at least 3 to 5.5 years to within this age. And if young juvenile spider requires at least 1.5 to 2 years to reach the larger size, and given the fragmentary growth data available, then this data suggests that the immature females of Euplus grandis require somewhere in the vicinity of 5 to 8 years to reach maturity. And this data is correlated to similarly Gaius Philosus spider, which the maturation requires at least 8 years in the wild. And we came to the conclusion. From the data, we can conclude that First, Euplus grandis is the new largest species in their genus. It inhabited the endemic part in eastern Australia. Second, they have been known to exhibit maternal care, where the babies live together with the mother until they disperse. And the third, they have been known to be able to repair the broken trapdoor with the new one within some times. And the mortality rate of this species is high in young stage, but decreasing once they reach adulthood. In general, Euplus grandis reach the maturity stage in 5 to 8 years in the wild. And I'm gonna tell you, this data is hard to impossible to obtain without using continuous research in slow science method. It's very important because to know this a lot of data, we cannot do some how to say some short time research. We have to research it in a lot of time. Like you can just make it from the first month into this other month and get some instant result. No, you can do that. And once again, that's how the slow science take part in this research. It's very important to know this all of this data. And we also provide some possible future methodology. The first one is to use the next generation sequencing or NGS to make individual genetic assessment, like provided by Su et al. In 2015. They said silk were potentially used in NGS, but the successful rate still unknown. And the second one is the environmental DNA or eDNA to estimate gene flow and the genetic variation. But once again, this methodology requires further testing. And for the conservation implication, we may be asking, why do we spend a long time just to understand a particular species? So the answer is, with upcoming changes in natural habitat, we have to know how species characteristic to overcome such issue. And by understanding it, given that conservation planning and management are at best hampered and at worst prevented. And once again, uh, I'm telling you that we have to know how particular species live, and by using short time experiment we cannot get the best result. We have to do it from time to time. We have to look into their life, how they live, how they interact with the environment, and that's how we get the data that important to prevent and to implicate the conservation for them to having a better life in the wild. So this is the end of my presentation. Thank you for watching. If you have any question, feel free to ask in the comment below. Thank you.